The topic of discussion will be on growth factors that would aid in the periodontal regeneration. As we all know, periodontal diseases constitute the inflammatory condition marked by the loss of attachment apparatus in response to microbial stimuli. So this periodontal disease caused by the microbial infections results in the loss of attachment apparatus, apparently the bone destruction when not treated, leading to tooth loss. Many treatment options are available, which includes the non-surgical, surgical and regenerative procedures. These regenerative procedures aim at achieving new cementum formation, new alveolar bone formation and functionally oriented periodontal ligament fibers. So there were certain polypeptide molecules released by various cells in the affected areas to regulate events like wound healing and tissue regeneration. They were called as the growth factors, which are a family of polypeptide biological mediators which modulate cell responses such as cell adhesion, survival, proliferation, chemotaxis and differentiation. How do they act? They act on a specific cell surface tyrosine kinase receptor which are present on target cells like cementoblast, osteoblast, periodontal ligament cells and fibroblast, which then activate the second messengers of terminal effectors to evoke a biological response. There are various types or classification of growth factors. Include platelet-derived growth factor with their isoforms from AA, BB, AB, CC to DD. Vascular endothelial growth factor from A to F along with placenta-derived growth factor. Transforming growth factor beta superfamily, which has the transforming growth factor beta, inhibins, activin, anti-Mullerian hormone, bone morphogenic protein, dicapentaplasmic, VG1. Epidermal growth factor, which include the transforming growth factor alpha, Schwannomo-derived growth factor, heparin binding epidermal growth factor, beta-selenin, epiregulin, neuregulin. Fibroblast growth factor from 1 to 23 isoforms and insulin-like growth factor isoforms 1 and 2. Hepatocyte growth factors include the macrophage stimulating protein, then macrophage and granulocyte colony stimulating factor, which also include the interleukin 3 and erythropoietin, and neutrophil family, which includes the brain-derived neurotrophic factor and neutrophil 3 to 6. There are various functions of all of these growth factors, but particularly we can highlight certain functions which enhance the periodontal regeneration. Growth factors stimulate cell proliferation. Example, platelet-derived growth factor. They enhance function of cell and cell differentiation. Example, bone morphogenic protein. They stimulate matrix synthesis. Example, transforming growth factor beta. They act as a cofactor for gene expression. They have a biomimetic property occurring during embryonic and postnatal development. They have a natural reparative process when a combination of growth factors was given. Then the single recombinant growth factor also induce molecular, biochemical and morphological cascades that will result in tissue regeneration. With the advantages of growth factors, there are certain disadvantages that is, they can be produced only by certain cells and can be stored locally, and their half-life is very short, that they have to be provided in very higher concentration, and when given in higher concentration, there occurs a cell death upon withdrawal, and they require a particular delivery system for the sustained release of the growth factors in lower concentration and also to give a cocktail of growth factors. Coming to the recent advances, one is regarding the regulation of stem cells by growth factors. Another is the usage of platelet concentrate, which would have the growth factors. So this regulation of stem cells is by the human periodontal ligament cells, which contain the mesenchymal cell population that persists in adults. So during an injury, the growth factors like bone morphogenic proteins and platelet-derived growth factors recruit these mesenchymal cells. This was confirmed by means of a study by using a sonic Hedgehog genes, which is nothing but a protein which would help in the embryogenesis and organogenesis. So when they stained positive for the antibody, then it would say that it has an increased cell proliferation. Coming to the platelet concentrates, it is a source of autologous growth factor for periodontal regeneration. So Marks and co-workers describe the use of autologous platelet concentrate to modulate bone healing. So these platelet-derived growth factor consists of certain isoforms of platelet-derived growth factor AA, BB, AB, and two forms of transforming growth factor beta 1 and beta 2, vascular endothelial growth factor and fibroblast growth factor 2. They have functions in wound healing and can be used in therapeutic approaches like sinus floor elevation, bone augmentation, mandibular reconstruction, maxillary cleft palate regeneration, and periodontal defect regeneration. So, all of which have the action of cellular migration, myofibroblast proliferation, extracellular matrix production, wound healing, ultimately periodontal regeneration. So, to conclude, the objective of administering growth factor in the treatment of periodontitis is to mimic the normal developmental process and enhance normal wound healing response to promote complete regeneration of all attachment apparatus and aids in tissue regeneration. Thank you.